This is lecture 11 in Principles of Metabolism, and in this lecture we'll look at some major classes of enzymes. So we said in the introduction lecture that even though metabolism looks very complicated and consists of a large number of enzymes and reactions and pathways, there are actually only a few classes of enzymes and a few types of metabolites. So we'll look at some of the classes of enzymes that constitute metabolism. Pretty much all enzymes in metabolism can be put into one of 6 major classes of enzymes. So these are the classes shown here. This is a system that was put together by the Enzyme Commission, and this is a kind of panel. It's published by the IUBMB, the International Union of Biochemistry and Molecular Biology. And there's this website shown here where you can explore these classes. So we are biologists after all, so we try to classify everything into trees, and this is no exception. So there are these major 6 classes, but then each of those have subclasses, so there are kind of specializations. There is a sub-subclass, and then within each sub-subclass you can finally find an enzyme. So it can be a structure looking like this. You can have the major class 1, the oxidoreductases, and then the second group is whether it acts on alcohols or on aldehydes, or what it acts on. And then in this case, the sub-subclass would be what kind of electron acceptor is used. Is it NAD? Is it cytochrome, or what is it? And then you get to the actual enzymes. So it's kind of a taxonomy of enzymes. So let's look at some examples from each of these groups and see what kind of enzymes there are. So starting with the oxidoreductases. So these are the redox enzymes that we have seen in previous lectures. So pretty much everything that does oxidation reduction reactions is in this group. So typically enzymes that do oxidative metabolism, like in this case lactate dehydrogenases, which oxidize lactate and into pyruvate and use NAD as an electron acceptor. That's a typical example of an enzyme in this group. So here you will also find the biosynthetic enzymes that use NADPH, for example, as an electron donor to drive anabolic reactions. So that's the oxidoreductases. And these enzymes typically involve an exchange of energy between a substrate and a carrier like NAD. So transferases are enzymes that transfer parts of metabolites. So an example is transketolase, shown here. This is an enzyme we encountered in the pentose phosphate pathway. So what this enzyme does is that it takes this 3-carbon skeleton of glyceraldehyde, and it uses that to form part of a 5-carbon sugar cellulose. And at the same time, it takes these 2 carbons from the 7-carbon sugar C2-heptulose and sort of joins those to the other 3 and forms a 5-carbon sugar. And then the remaining 5 carbons of C2-heptulose are going into ribose. So it forms 2 5-carbon sugars from 1 3-carbon and 1 7-carbon. So this is a typical kind of cut and paste type of reaction that the transferases catalyze. So this particular enzyme is energetically neutral. There is no ATP or NAD or anything like that driving this reaction. And that kind of makes sense because there's similar sugars on both sides. There are some instances in the transferase group. For example, the kinases are here because that's transfer of phosphate groups. Those break bonds that are energetically sort of high energy. So some of the reactions in this group are driven by high energy bonds. The hydrolases are enzymes that cleave metabolites using water as a reactant. So there's a lot of these, and that kind of makes sense. There's a lot of water in cells. It's the major solvent, and so evolution has probably had a lot of opportunities to come up with reactions like these. So this is a typical example. This is a glutaminase. So in this case, water is used to attack this amide group, the nitrogen part here at the end. And so this gets substituted now for an oxygen. So this becomes a carboxyl group. And the amide group leaves as ammonia in this case. So this is a case where we are cleaving off parts of a molecule and we are using water to do it. And the water typically joins and forms a group like this. So hydrolases also include the phosphatases. And ATP hydrolysis is one example of a hydrolase type of reaction. So many of these reactions are energetically favorable and spontaneous. Glutamine, for example, is a case that this reaction actually happens without enzymatic catalysis even on the time scale of a few days or a couple of weeks. At body temperature, glutamine actually decomposes. So this class of enzymes accelerate reactions that usually happens at some appreciable time scale already in water. The lyases is a family of enzymes that also cleaves molecules, but this is fundamentally different because the lyases cleave molecules without using water, which means that there is no addition of water into these molecules. So a classical example is the aldolase that we have seen in glycolysis. So this cleaves fructose 1,6-bisphosphate by breaking the bond over here. And in this process, it forms two triose, three carbon sugars, the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So lyases are different from hydrolases in this respect, that they can cleave molecules without using any additional reactant. Isomerases, as the name suggests, are enzymes that convert between isomers of metabolites. So these are enzymes that can change the stereochemistry, configuration, like change an D amino acid to an L amino acid. There are enzymes in this group that move certain parts of a metabolite around a bit. But total, these enzymes don't add or remove anything from metabolites. They just change their form. So here's one example. This is glucose 6-phosphate isomerase. So what this enzyme does is that it changes a bit of the structure around here so that the glucose sugar bound to a phosphate here becomes a fructose sugar. So there's a carbon that's originally in this position in the ring that now so sticks out from the ring in the fructose form. These enzymes are typically energetically neutral, so there are things that are close to equilibrium. So when we see isomerases, we expect that these are not driving the reaction in any particular direction or extracting energy or anything like that. The last group are the ligases. So these are enzymes that join molecules together, form new bonds. And in this group, we have things like CoA ligases, enzymes that take cofactors and attach metabolites to them. DNA ligases is if you've ever ligated together a couple of nucleic acids in a vector, that's also an example of a ligase. Ammonia ligases, adding ammonia groups to metabolites, is also an example of this. So ligases usually they form new bonds, and that is usually something that requires energy. So typically ligases are driven by ATP. They usually require energy to carry out their function. So when we see ligase type of reactions, that's usually something that's energetically costly. So those are the 6 major classes of enzymes. And if you know about these classes and roughly what type of enzymes they are, then you actually know about most enzymes in all of metabolism. because metabolic pathways and metabolic networks are just combinations of these types of enzymes. So we're mixing and matching different types of enzymes to carry out pathways and transform metabolites. So here's an example. This is the serine synthesis pathway, starting from the glycolytic intermediate phosphoglycerate. So this pathway starts off with an oxidoreductase, does a reduction, and changes this hydroxy group into a keto group in this case. Then it uses a transferase to put an amine group onto this piece of the carbohydrate to kind of make an amino acid. It's a phosphoamino acid at this point. So now we want to get rid of the phosphate group, and then we use a hydrolase. So the hydrolase uses water, and the phosphate group comes off, and we have serine, the product of the pathway. And so when looking at metabolic pathways, and if you use human-seq, as in this example, you will usually find in all these metabolic databases the EC numbers down here. So you can see what kind of classes of enzymes show up in these different steps. And I think this helps a bit to understand how the pathways are organized and which steps are energetically demanding, which ones are just a transfer of a group, which ones might be spontaneous, and so on. So this is a useful, generally applicable concept, I think. You will encounter sometimes some enzymes that perform something that doesn't look like anything that fits in the EC system, something that looks more complicated. And usually those are more complex enzymes that have evolved to perform several reactions. Commonly they are have multiple domains or are multimers of simpler enzymes. The pyruvate dehydrogenase complex is one example. So the overall reaction doesn't really fit in the EC system, but the separate reactions that make up the pyruvate dehydrogenase complex do fit into the EC system. So you may run into some examples where this doesn't fit easily, but in most of the cases, this is a very good system for understanding metabolic pathways.